Hello and welcome to the sixth and final installment of the 2016 INSAR Summer Institute. I'm Carolyn McCormick, co-chair of the INSAR Student and Trainee Committee and a postdoc at Brown University. The INSAR Summer Institute is a 6-course online series presented by the International Society for Autism Research. The goal of the Summer Institute is to provide a forum for the presentation and discussion of autism-related research topics that is accessible for people new to the field. The Summer Institute was originally motivated by and conceptualized for early career researchers, specifically graduate students and postdocs, but it's open to all those interested in autism research. We hope that the Summer Institute provides an opportunity for scientists with different backgrounds and from around the world to learn from each other and ultimately advance our understanding of autism. The Summer Institute sessions are free for everyone to attend live and available to INSAR members for replay through their MyINSAR account. This is the second year we're offering this training opportunity, and the theme for the presentations is familial aspects of autism. Today's session features a presentation from Dr. John Constantino, who will be talking about the broader autism phenotype. Know that there are background materials for today's talk. If you haven't already, you can download these now at autism-insar.org. These materials define some key terms and provide suggested reading in the literature. As with all of the Summer Institute sessions, a group of trainees worked with Dr. Constantino to prepare these course materials. The working group was led by Christina Cottle, a doctoral student at the University of Utah. Other members of the working group who contributed to today's session are Lauren Friedman, Elise Hayter, and Joshua Page, all from the University of Washington in St. Louis. Before we get started, know that we welcome questions from the audience. We will leave lots of time for discussion during the second hour of the session. Ask a question anytime during Dr. Constantino's presentation by using the chat window on the left of your screen, and we'll get to as many of them as possible during the discussion period. At the very end of the session, we will leave a few minutes to talk about career development. If you want to get some advice about career development topics from Dr. Constantino, please post your questions in the chat window and we will incorporate them in our discussion. We also greatly appreciate feedback from attendees. When you end your session, please take a few moments to tell us about your experience today in the comments window. Now, I would like to hand the session over to Christina Cottle. Thanks so much for joining us today, and welcome, Christina.
Thanks, Carolyn. Before we begin, I want to say a huge thank you to Carolyn McCormick for organizing this year's Summer Institute. She's done a fantastic job recruiting wonderful speakers and keeping us all in line and making sure we get done what we need to get done. So thank you, Carolyn. I think it's been a great Summer Institute. I also want to thank Lauren, Elliot, and Joshua who helped me create the pre-session materials. You're a fantastic team and should definitely be recognized for their hard work on this. I'm now going to introduce our speaker, Dr. John Constantino. He is the Blanche Ittleson Professor of Psychiatry and Pediatrics at the Washington University School of Medicine in St. Louis. He received his medical degree from Washington University School of Medicine in St. Louis, and then he completed 2 residencies, one in pediatrics and one in psychiatry at the Albert Einstein School of Medicine. He later completed a fellowship in child psychology. Dr. Constantino is currently the director of the William Greenleaf Eliot Division of Child and Adolescent Psychiatry. He is a principal investigator of an NICHD Intellectual and Developmental Disabilities Research Center, and he is also psychiatrist-in-chief of St. Louis Children's Hospital. Dr. Constantino's work has focused on understanding genetic and environmental influences on social development in childhood. Along with a very established team of researchers, he has developed an advanced method for measuring autistic social impairment as a quantitative trait, allowing for patterns of transmission of autistic syndromes and subclinical autistic traits to be traced in families and populations. These findings from the studies have significant implications for gene discovery, identification of new targets for biological and developmental therapy, genetic counseling, and preventing impairment and adaptive functioning that is incurred by autism and other disorders of social development in childhood. And he'll be speaking to many of these things today. So, Dr. Constantino, I'll turn it over to you.
Thank you very much, and good morning, everybody. Thanks so much for dialing in, and over the next hour, I'm looking forward to talking a little bit about the broader autism phenotype. This particular topic is one where I think if I had to summarize it, I would just say that there's more there than meets the eye. I remember reading Joe Piven's seminal paper on this in the American Journal of Psychiatry as a probably as an early career investigator. I can't remember what year it came out now, but recognizing that there existed among the families affected by autism, and this had been observed by others as well, manifestations of the autistic syndrome that were not the full syndrome. And once this term was coined, I think the next step in the field was to try to characterize it, measure it. It had been very sort of advantageously picked up on the basis of different kinds of family history interviews and so forth of family members, but refining the measurement and implementing a refined measurement in large populations has taught us a significant amount about the biology of autism. And so I hope to kind of tell that story today, and we'll look forward to dialogue or questions or discussions about that as we go along. So I'll first just disclose interest for everybody. I do a little bit of industry consulting, and just in the past year for Roche Pharmaceuticals. I don't have any stock equities. I will talk about the scale that we developed, the social responsiveness scale, for which I receive royalties from Western Psychological Services, the publisher of the instrument, and that's been really a private-public partnership from the beginning that's allowed the scale to be translated into now over 30 foreign languages and to be really implemented around the world for relatively inexpensive use in populations, including public health and developing countries. I receive research support from NICHD and the U.S. Centers for Disease Control for our autism research program. Those are current active grant sources. And I also receive grants, and maybe we talk about this in the end, for a couple of programs that are that subsidize, they're governmental subsidies for what I would call clinical and research hybrids, and they allow us to actually not only study patients but take care of them. And I think that's been a really important theme of my own career. And again, in the end, especially for the young investigators, I'm glad to talk a little bit about how that's come together and why I think that's been so important. So why is the broader autism phenotype important? Well, just to kind of whet your appetite and let you know what I'm going to try and talk about today. So first of all, I think it's taught us something about the genetic structure of autism, so that tracing patterns of autism symptoms in families has given us clues to the genetic structure and the biology of autism, which I'll talk through today in ways that, again, don't immediately meet the eye. Studying the broader autism phenotype also advances the concept of an endophenotype that I'll define a little bit later in the talk, but that really identifies particular components of a disease syndrome that might be carried by a family member, particularly for inherited conditions, and that when combined with other factors then contributes causally to the origin of a condition, and identifying what those core elements that collide or converge in the development of a neuropsychiatric syndrome becomes very important. The clarification of the concept of comorbidity has been in some ways sort of a sleeper effect of studying the broader phenotype because it turns out, I think as probably most of you have encountered, that autism travels also with other behavioral disabilities and psychiatric syndromes. And so sorting out what is attributable to autism and what is attributable to other types of neuropsychiatric liability becomes a very important issue in understanding in any individual patient or in groups of patients what's going on, and understanding that even a subclinical component of autistic liability can actually have extremely important consequences when coupled with another psychiatric syndrome. So the point there is that almost any child behavioral syndrome whether it's an anxiety disorder or an obsessional syndrome or depression or psychosis or any kind of psychiatric syndrome, if you add even subclinical autistic liability to that, which can be measured when you measure the broad phenotype, generally that is associated with a higher degree of functional impairment as a result of the primary disorder. So that even though that child doesn't have quote unquote autism, when they have a condition that is accompanied by significant overlay of autistic-like traits and symptoms, that has really important implications for how severe that other condition actually is and how much they're impaired. I think the study of the broader autism phenotype also yields insights for all of human social development, and I'll get into that as we go along. But just a preview of that is that again, the forces that cause autism, the genetic influences on autism, appear to operate through the entire population. And so it's not studying the broader phenotype, is really studying a dimension of social competency that runs through all of human nature and all variation in social development. And as a kind of a quirky example of how that affects human beings, one thing I talked about was this issue of comorbidity even for subclinical states. But another one that I'll talk about today is that it's essentially that that variation in this particular phenotype, the way it lays out for the human population, actually explains somewhere around 10% of all the variation in mate selection. How people, select the people that they mate with is attributable to variation in this particular phenotype. So that has also important implications from a population genetics perspective, risk aggregation across generations, and also just, how, mate selection occurs. Also, risk prediction in future generations. I'll talk about that and why the broader autism phenotype can actually index much more risk than what it looks like in an individual adult, especially with respect to the implications for offspring outcomes, and then implications for how do we really diagnose autism. What does it mean, given what we know about the broader autism phenotype? How does that feed into our current understanding of what does a diagnosis of autism even mean? And try to get into that a little bit as well. Okay, so I spent a lot of time on sort of giving a grand overview there. I hope that will at least give you some idea of what the rest of the talk will be like, and that it might be worth hanging into the webinar. So I'm glad for those of you who are with me. And although those seem like a lot of different sort of eclectic topics about the broad phenotype, one way to think about this, and the way I'll tell the story today, is that I think most of what we learned and most of the implications of understanding deeply what that broad phenotype is all about comes from what I'll call a tale of three distributions. And everything on this particular slide, if you take it to its logical conclusion, is essentially spirals into what we've learned about the biology of autism. Again, through all those different themes on the last slide, really can be traced to sort of three fundamental observations, and then follow-up studies that are based on those observations. So let me just talk through them briefly, and then we'll go through the rest. But I think this is, again, an important kind of take-home message, especially for the young investigators, that that sort of identifying a particular novel finding, even in something that seems sort of simple, and in fact, one of the things that we've done over the years is simply count symptoms in a quantitative way. I remember meeting a number of years ago Eric Kandel, Nobel Prize winner. I think that most of you know about his work in aplesia and the neural basis for learning. for which he won the Nobel Prize. And one of his things that he said in his advice to young scientists was pick one thing and study the hell out of it. And that was his advice, and I remember that, and it's stuck with me. And you know, when I began my work in counting autistic symptoms, recognizing after Joe Piven had published this paper and his colleagues on the broader autism phenotype, and recognizing that we didn't yet really have a way to precisely count and quantify nuanced manifestations of the broad phenotype, that the job was to do something that, as a psychiatrist and a pediatrician, I never thought that I would spend any time doing, but it was to develop a measurement strategy that would allow us to do this in a different way. And so we developed this measurement system, the SRS, the Social Responsiveness Scale, to quantify autistic traits in the full range that they are observable in populations. And so in this tale of three distributions, I think the most important things that we learned were as follows. The first was that when we just counted autistic-like traits and symptoms, and I'll get into a little bit more about how that measurement actually works, but when we just count them, what we see in the general population is a full continuous distribution, a slightly skewed bell curve, as it were, where you can see on the slide here, this particular distribution of scores is a general population sample where a higher degree of symptom burden is to the right, and the proportion of population, proportion of the population at each level of impairment is on the y-axis. And what you can see for both boys and girls, boys shifted a little bit more towards the pathological end, girls towards the non-pathological end, is this smooth continuous distribution where we took an instrument that distinguished autistic syndromes from other psychiatric syndromes, launched the instrument in the general population, and to our surprise didn't see that, oh, here were the normal children and then here were the affected children. Rather, what we saw was just this smooth continuous distribution where it's really impossible to tell where the cutoff lies between affectation and non-affectation. That was the first distribution. The second distribution was the observation of when we examined relatives. And I'll show a number of different takes on studying relatives. We've studied relatives in the same generation, so that's siblings. We've studied offspring. We've studied parents and grandparents. But in this particular slide, it depicts the situation that when we first observed the distributions for the brothers of boys. Here are boys with autism. Here are boys with other psychiatric conditions. And here are their brothers. And what you see among their brothers, again, is a smooth distribution of affectation, shifted to the right in comparison to the brothers of children with other psychiatric conditions, and mimicking, in a way, this condition, this smooth, continuous distribution of autistic traits. In this situation, this is for children with autism spectrum disorders. So you can, if you were to place this on this particular scale, this whole group lies in this tail of the distribution. The brothers actually are anchored in normality, but fan out into a broad distribution, the mean for which is much higher than the mean for boys who are brothers of children with other psychiatric conditions. And this taught us that the aggregation of these traits in families, and we've seen this over and over again, occurs in a quantitative distribution, not an all or nothing kind of distribution, where this particular broad phenotype becomes very difficult to tell which of the brothers, have any trace of the condition or which ones don't, because their distributions are as quantitative as children in the general population. These are the brothers. In the third panel here, the third distribution that taught us something very important about autism from the broad phenotype involves the sisters of children with autism. And it turns out that there's a significant difference in the distribution, particularly in families where autism is known to be inherited on the basis of multiple family members being affected. That in contrast to this situation that you see with male relatives, which is this smooth, continuous distribution that's just sort of shifted to the right in comparison to what a normal distribution would be. The situation for girls is interestingly somewhat different. The sisters of children in multiple incidence autism families show a distinct bimodal distribution, where in some ways it's as if the girls are either affected or they're not, in ways that the brothers take on the full range of symptomatology, where it's impossible to differentiate a group that's carrying or affected by some aspect of the syndrome. The girls, there's this notch in the distribution. We've seen this in 3 replicated studies, multiple very large sort of clinical epidemiologic studies, and particularly in the multiplex families, the families that have multiple individuals affected. And there's this remarkable notch in the distribution that I won't go into all the detail now, but what tells us, and I'll talk about this later in the presentation, that tells us that there may be something very special about females that results in this notch in the distribution, and that may be the actual core of why it is that autism universally across cultures, across all populations, manifests essentially a 3 or 4 to 1 sex ratio. And I bring that up because historically it had been assumed that the sex ratio in autism was what it was, 3 to 1 or 4 to 1, because autism worked just like an X-linked intellectual disability. And that, someday, this is what people used to think, that someday when we find the genetic causes for autism or the biological causes for autism, that many of them will be X-linked. Well, it turns out now that we have a handle on knowing not all, but many of the genetic liability and influences on autism and where they lie in the genome, that essentially almost none of them are on sex chromosomes. And so that raises, in the recent 5 years or so, when we started to be able to identify where inherited liability to autism resides in the genome, and I'll say a word about that in a minute, but the fact of that new discovery has been that it's not on sex chromosomes, and that really raised the issue of, if it's not on sex chromosomes and autism is a fundamentally inherited condition, how on earth is there a 4 to 1 sex ratio? And we think that this particular distribution gave us really important clues into what exactly was going on there, and I'll get to that towards the end of the presentation. Okay. How do we know that autism is inherited in the first place? So I'm going to sort of move through the rest of the presentation in a manner that in some ways is going to carry a sort of a fundamental assumption about autism, that it is primarily, not exclusively, but primarily an inherited group of syndromes. And the reason why we think that it is an inherited group of syndromes, I want to cover that briefly because I think it predicates all of what we're talking about here in the broader phenotype. And I'll say that, even if a condition is inherited or as a function of single or multiple genetic factors, obviously those genetic factors always interact with the environment. And I'll show a way in which we think that that's happening towards the end of the talk. So for me to say that most of the causal variants in the development of autism, the origins of autism, are inherited or genetic in origin doesn't mean that I think that there's no role of the environment. It means that inherited liability or genetic liability is one of the most important factors in its cause, and that then those factors particularly may be either correlated or interact with usual human environments to result in this particular syndrome. Well, why do we think that? Well, the reason why we think that is that in studies now that really honestly are totaling over 4 million human beings that have been studied in genetic epidemiologic studies, The identical twin concordance rate for autism is greater than 90% and probably closer to 95%. If you take all comers of sort of manifestations of the clinical autistic phenotype. And so that identical twin concordance rate could be attributable either to genetic or environmental factors, because identical twins, these are by and large studies of twins reared together. That concordance could be on the basis of either genetic factors or environmental factors, because twins share 100% of their genetic variation, and that's identical twins, and they also share their rearing environment. So it could be either. But what happens when you keep the rearing environment equal between the twins, so not changing that. So in the dizygotic twin situation, they're both reared in the same environment. That means they have the same mom. That means they shared the same uterus prenatally. That means they're exposed to the same pesticides. That means they're exposed to the same air pollution. It means that they're exposed to the same foods and the same vaccinations by and large. And so when you look at those children where the environment between the twins is kept equal because of their rearing together, but you cut the genetic similarity between the two twins in half, the concordance rate plummets. And that is one of the strongest pieces of evidence for the inheritance of autism that we have. And that's been replicated over and over and over again in most twin studies that have looked at autism, that this particular concordance pattern occurs. This recurrence rate is also true for non-twin siblings. So when siblings are staggered in time in a family and not having exactly the same environment, that particular concordance doesn't erode very much and still is sustained at about the same level. And all of this in comparison to the general population risk. About 60% of the cases of autism are sporadic, and that can be on the basis of what are called de novo or germline mutations. That means neither mom nor dad has the mutation or has the genetic variant or variants that are responsible for an autistic syndrome. And in either the sperm cell of dad or the egg cell of mom, that there was a mutation that did not match the genome of the rest of their bodies, but was unlucky enough to be transmitted to the fertilized egg that produced that child, and that's a germline mutation. And those de novo mutations are, we're learning more and more about how commonly they cause autism, probably from a standpoint of structural rearrangements in the genome. It explains about 5% of cases, and from the standpoint of sequence variants, just one or two nucleotide changes that change the actual function of a gene, that number can be somewhat higher, and yet we also know that there's going to be an upper limit to the number of de novo cases, because you would never see this pattern of inheritance necessarily if that gets too high, primarily because this pattern of inheritance extends also to half-siblings, and I'll explain what I mean by that in a minute. In other words, these particular de novo mutations are also shared by identical twins. And so it's possible that a lot of the inheritance pattern or this notion of inheritance could be inflated if there's a really, really high proportion of de novo cases that are inflating the identical twin concordance. So if, let's say if 60 or 70 or 80% of all of autism was caused by de novo mutations, then we would not have such an idea that it would be so strongly inherited. One of the ways that we actually know that that's probably not the case, that the de novo rate is probably much smaller than that, is because if you look also at the situation for half-siblings, what you see is a similar kind of cutting down in the risk. And so this gradual stepping down when you go from a full share of genetic similarity to, which could be falsely inflated, to half genetic similarity, that same decrement in concordance happens when you go another step to half siblings. I'll show that in a minute. So the point is that these estimations about heritability that are based on twin studies are really quite robust. 40% of the time, autism is patently familial. And so sometimes sporadic cases occur not because of de novo mutations, but because there just haven't been enough other children in the family to show another case of autism. For familial cases, you see autism in not only the index case, but in family members. And in either of these, in this particular situation, the mechanism of transmission could be highly polygenic, or there could be rare inherited variants that are transmitted to multiple siblings. And again, on this side of the ledger, on the sporadic cases, rare inherited variants can also show up just in one single member of the family, or the wrong combination of rare inherited liabilities can show up in one member of the family and not be manifested in other children of the family. So I just want to make sure that we're kind of predicating this on kind of an understanding of why do we think that autism is so strongly inherited. Here in the next slide is a study of half-siblings, and just to continue to make this point that when you compare full siblings, in this case in the Ian Registry and in a Autism Center of Excellence Network study, full siblings, if you take a very conservative categorical diagnosis of autism, in these particular studies, you see a concordance rate for full siblings on the order of 10%, and then that gets cut to about in half in the condition for maternal half-siblings, and interestingly even lower for paternal half-siblings. But again, it represents the idea that a very substantial share of what looks inherited through twin studies is not an inflation on the basis of the de novo mutation rate, and that in fact that the way the inheritance patterns for autism are operating, gradually being cut down in the situation between identical twins, non-identical twins, half-siblings, and so forth, is that these transmission patterns are highly suggestive of additive genetic influences for most of the public health burden of autism. And in fact, this is what was identified subsequently in molecular genetic studies, where most of the genetic risk for autism appears to reside with common variation. This is a seminal paper that was published in July of 2014 by Gogler et al., big group of autism genetics researchers, that indicated that when you look at common variants in the genome and how they contribute to autism risk in a large population, that over half of the variation is attributable to variation in common variants. And each of these variants explains only a tiny component of the risk for autism. And so, but this was one of the first molecular genetic demonstrations that autism is not only highly heritable, but a function of the aggregated influence of multiple genetic factors that accumulate in an individual to produce the syndrome. And this makes perfect sense in terms of why there would be a broader autism phenotype, why autism would be quantitatively distributed in populations, because if most of the burden is a function of these polygenic risks, then one would expect that in families where these risks are at higher levels in an index case, that other family members may have varying levels of burden of these particular symptoms. One of the things I want to take a quick pause to mention about this issue of common variation, and especially for the young scientists, is that one of the great motivations for studying the genetics of autism in the first place was to identify particular single gene causes of autism that, if they accounted for enough of the population attributable to risk for autism, and we could chase down the biology of those single genetic factors, that then we would have a better idea of how to develop biological novel biological kind of therapies that would be based on those individual genetic pathways. And the problem, of course, it turned out was that two problems. One was that any of the monogenic autistic syndromes the single gene syndromes were rare and only would account for autism in a small number of the total number of families affected by the condition. So even if we, let's just say, found a cure based on, let's just say, Fragile X syndrome, which we've known about for a very long time, that as a cause for intellectual disability and autistic syndromes, that if you could target it and treat it, you would still only cover a very tiny percent, less than 1% of all the autism cases. And so initially in genetics, there was the hope that there would be, genetic factors that would cover, sort of, as it were, more of the population. Well, that turned out to not be the case, that many families have autism on the basis of, sort of that family's own weird, rare way to autism, and that there's hundreds of pathways to autism, it looks like, not only from the standpoint of those families who have major mutations that are, in brain genes that are producing autistic syndromes that are individually, rare pathways, but also looking at this slide in the sense that most autism is caused by the aggregation of tiny, tiny components of risk that aggregate and produce the syndrome. And I want to just say that I think some folks might think that this should, make us feel, or at least give us pause, that, oh gosh, if autism is hopelessly polygenic, we'll never have, kind of a parsimonious biological target to treat it. And I just want to offer the example of another condition that's just as polygenic and just as heterogeneous, and that's attention deficit disorder. And yet, we do have biological targets for that condition that are remarkably effective. And just to note how effective they are, I always like to kind of point out this paper, and this is a little bit of a divergence, but attention deficit disorder is genetically very similar to autism and has a lot of overlaps with autism. And this is a paper that was published a few years ago that took the Swedish National Registers and cross-referenced, their registries are remarkable, and cross-referenced the adults in the Swedish National Register who were known to have a diagnosis of ADHD, and cross-referenced their records with the pharmacy records of when were they taking medications to treat ADHD and when were they not taking medications to treat ADHD, and then in turn cross-reference that with the records for crime, for the criminal records. And in every category of crime, if you were an individual with ADHD, the likelihood that you would commit a crime was much greater, was significantly greater when you were not taking a medicine for this condition rather than when you were taking a medicine for this condition. And I offer this in the sense that this is a very powerful demonstration in the sense that even for a polygenic syndrome, a biological target can be derived, a treatment can be delivered, a treatment that can be effective across numerous different pathways to the condition. We know this for a fact in this particular condition. And to have the kind of public health impact that was demonstrated in this study, and this definitely needs to be replicated, but I think it stands as a sort of shining example in a sense of what's possible, even if autism is very messy and polygenic and distributed in nature in this way. If that's the way it is, we have to, deal with it and approach science in a way that that's going to honor those facts about the condition. Okay, related to that, I want to point out just an interesting study that looked at even those individuals who are affected by de novo mutations. This is a group. This is a study that was that examined a group of individuals in a registry that involved multiplex autism, so multiple family members affected. And they asked a very simple question. If one of the members of the family was affected by autism on the basis of a de novo mutation, when or a potent kind of structural rearrangement of the genome that covered significant brain genes, how often did that that particular potent mutation occur in a sibling. And it turned out that only about half the time in families where two siblings have autism and you identify a deleterious variant in sibling number one, only half the time was the deleterious variant that was identified in sibling number two was either there or the same variant, or oftentimes it was a different variant. And it was interesting how this particular study was interpreted, as if, you know, that the collection of autism in families is because lightning sometimes strikes twice, and, you know, autism happens because of a high, you know, random rate of these mutations across family members, which actually is true, that there can be different mutations in family members. But what wasn't so much understood, I don't think, from the outcome of this very important study was that the nature of the concordance between the siblings had to do not with the particular genotype for that particular mutation, but with the fact that running in that family is a background of risk for autism on which those mutations were superimposed. And what we think now, and I'll show this in great detail in a minute, but what we think now is the way we've learned a lot about the very way a mutation works, that it's not the sole cause of a condition or a deterministic factor unto itself, that it always operates on a genetic background. And that genetic background can be indexed by studying the broader phenotype within that individual and family and understanding what exactly happened and what was the role not only of a particular genetic hit that may explain part of the affectation in a family member, but also how the liability for other family members is actually traveling in the family. Well, so as I said before, to do this, we had to develop a method for studying quantitative autistic traits. And many of you are familiar with this particular instrument, the social responsiveness scale. And essentially what it does is it counts symptoms of autism on the basis of naturalistic observation of children as young as 30 months of age and as old as 99 years old. So from 30 months to adulthood, on the basis of the observations of untrained raters, adults who know that individual and can, in 15 minutes, rate them according to this scale with respect to how they behave in interpersonal natural contexts. And the long story short about this particular scale is that when you look at particular populations, let's see, one second, I think I lost my, here we go. Need to change my slide. There we go. This is just one of many, many examples when you compare, in this case, individuals with autism to their unaffected siblings. This is from the Simon Simplex collection. You can do this in comparing individuals with autism with other psychiatric conditions. But what you see is that for both males and females, the scores on this instrument are about 3 standard deviations higher for individuals with autism than for other individuals in the population. And so, let me just make sure I didn't, yes, sorry about that. And so, as I showed in the tale of 3 distributions, when you look at this particular counting of nuanced symptoms and traits that differentiate individuals with autism from those that don't have it, and you launch that in the general population, what you find is this smooth, continuous distribution. And this happened to be a distribution, a sample of twins. And one of the other things that we learned right out of the blocks from studying twins in the general population was that the heritability for these particular traits was as high as was conceived of as for autism itself, which was very interesting. These are not individuals with autism. These are individuals in the general population who are being measured on an autism-based scale for reciprocal social behavior. This is not the only scale where this has been tried, and these are 2 other examples of the study of quantitative autistic traits where general population samples were used. So this is a study by Angelica Ronald and her colleagues in the twins early development study in the UK that used another quantitative trait scale, the CAST, to count these symptoms, and showed very similarly, at least on the pathological end, a smooth, continuous distribution within the general population. And likewise, David Skuse using the SCDC showing the same kind of continuous distribution in a general population sample. Interestingly, in the TED study in the UK, those were also twins in the general population, and using a completely different kind of instrument still derived the exact same, almost exactly the same level of heritability for these traits in the general population. These particular traits are stable over the course of time, both in clinical samples and in the general population, so that when you measure these quantitative autistic traits, which map to the broader autism phenotype, which map to social variation in the population, where a person stands at time 1, they tend to kind of stick there at time 2. These are baseline ratings of school-age children by their mothers, and this is a follow-up over years of time in a second wave of follow-up. And what you see is that the ratings by their mothers really don't budge very much on average over the course of time. This is a scatter plot of what these ratings look like over years of time. And this is a spaghetti plot of the sort of continuity over time of a group of children with autism who vary quite a bit with respect to, the total range of deficiency. But when you look at them, essentially these are individual tracings of kids over the age span from about 3 years of age to about 20 years of age from a longitudinal study. And what you see is this sort of consistency in a spaghetti plot, this sort of dense area of where the life course of children with autism lie on serial ratings over time that are the essence of how these trajectories were mapped, and these are their unaffected siblings over time. And these can be kind of cleaned up statistically to show the kind of life course of boys with multiplex or simplex autism, and girls with simplex autism, girls with familial autism, children with psychiatric conditions that are not autism. And interestingly, they start off a little elevated and tend to droop over time, so normalize over time in that particular group. And then these are unaffected sibling controls. Now, I showed these plots that would map the ratings over time by trained raters using a checklist instrument. And recently, we've attempted to move back the measurement of these traits earlier in development down to the age of 12 to 18 months, and have been very worried that it'd be very difficult for parents to make sort of nuanced ratings of children on the basis of items that are in a questionnaire. And so what we did was to develop another type of instrument in which a short video clip of a young child interacting with an examiner is viewed by the rater, and then after viewing this 3-minute video clip, or actually 2.5 minutes, which I'm going to show you, then the question is posed to the rater: compared to that kid, rate your kid, rate the child that you are rating in comparison to that. And this provides a video anchor, and I'll show you the results of that, but let me show you the video first, and I'll stay quiet during this video, that allows a parent of a young child to view what represents a very high degree of competency in social communication and reciprocal social behavior, even before a child has a significant command of language, of verbal language. So let me roll this clip, and you can watch that, and then I'll talk about it afterwards.
Okay, we can use the ball. The ball. Okay, we have to lock the cabinet back up. Can you throw it to
to Mom. Throw it to Daddy. Mom, my turn. I'm going to give it back to you. Yay! I got to go get it. You ready? I'm going to throw it to Paige. Ready? Catch. Boom, 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 boom. He's so funny. What do you like to play with the ball? Do you like to play catch? Do you like to catch the ball? Catch the ball! Can you go get it? Can you go get it? Can you kick the ball? Bye-bye. Bye-bye. They went bye-bye. I like that. Should we put these away and find something else to do? Can you help me put things in the bag? Clean up. You know what that means. Clean up. Thank you. Clean up. That's good. Clean up. Should we put Elmo in here? Should we put the plates in here? Plates. Good. No? Okay. I understand. Say hello. Hello. How are you? I have a blanket here. What's that? Paige. What is it? Bring the bunny over here. Yeah,
Bunny rabbit.
That's right. What's the bunny doing? Hop, hop, hopping? Is the bunny hopping? Is the bunny hopping? The baby likes the cake. Oh.
Made the baby
Thank
thirsty.
you. I think so. Thank you. That's a Celia.
So what you saw in this particular video was a child who was interacting, referencing, certainly pointing and declaring what she wanted, sharing her intention, reciprocally responding, gesturing, expressing emotion through facial coordination of eye gaze and facial gestures. And so what happens is then after the rater looks at this particular video, which is open access, which we want anybody to use that would want to use this particular scale. This is called the Video Reference Rating of Reciprocal Social Behavior. And when we implemented this particular measurement, and so after the viewing of the video, then there's a series of questions. There are about 10 video referenced items, give or take, that are directly referenced to that video anchor, and then a number of questions that are just questionnaire-based additional information. When this scale is used, again, what we see in a general population sample, this is a paper that was published by Dr. Natasha Maris last year, a similar among general population twins, a similar kind of distribution in the general population with a couple of outliers that indeed ended up to accrue a diagnosis of autism in this particular prospective study of young twins. And the heritability of this instrument is manifested by an identical twin correlation for these particular measurements on the order of 0.9 for both male-male identical twins and female-female identical twins, and then a non-identical twin correlation much, much lower than that on the order of 0.43 or 0.34. So seeing in some ways the same kind of pattern that we can pick up these quantitative traits earlier and earlier in life. I already talked about this particular aggregation pattern, and just to reiterate the point that the method for accruing and identifying these traits were related to this strategy for measurement of quantitative autistic traits in family members. And so I wanted to get to this issue of an endophenotype. And an endophenotype is essentially defined as a trait that's associated with an illness. It's heritable. It's state independent, but could require challenge or provocation in a systematic, standardized way. It exhibits familial co-segregation with an illness. It's found in some unaffected relatives, and in general that the aggregation of the trait is lowest in the general population, a little higher in unaffected relatives, a little higher in spectrum disorders, and highest in individuals who are categorically diagnosed with the condition. And so in this sense, these kinds of measurements, these quantitative autistic traits, which don't define autism in the general population, but define some variation in the aggregation of these traits, appear to be functioning as an endophenotype. And work by Elise Robinson and, again, Angelica Ronald and their group looking at large enough twin samples began to demonstrate that not only were the traits and symptoms of autism in the general population as heritable as autism itself, but literally caused by the same genetic factors. And in a large twin study where there's enough individuals in that study to be at the clinical affectation range of the distribution, you can ask the question of whether the genetic influences on individuals at the extreme overlap with the genetic influences. Even if you don't measure them molecularly, you can do this mathematically in genetically informative samples like twins. And in this particular study, they demonstrated that when people have affectation that's at the 5% extreme, so let's say you're at the instead of the 99th percentile and above, which we diagnose autism at about 1% of the population. If you're instead of at the 99th percentile, you're actually just at the 95th percentile, the causes of being at that elevation are really quite the same, overlap almost completely. And what that looks like varies from family to family, but generally represents just a lower level of trait severity across the characterizing traits and symptoms of autism. So whether in the realm of social communication and interaction, where it can range from, a relative disability or not being as facile in the area of social communication, or being extremely, let's just say, aloof or nerdy or quirky, are having a lot of difficulty even with motivation for a reward value of social interaction, all the way down to the opposite end of the spectrum, which is a high degree of social facility. And it turns out that the genetic factors that operate to determine one versus the other appear to overlap as well, that they're really controlled by the same types of genetic influences, just different allelic variations across the different genetic factors, the different genes that are contributing to these influences, which is really interesting from a human social development standpoint. And this kind of fact about the ability to, well, first of all, that these genetic factors are traveling within the families and that they overlap with the condition itself, then turns back to the issue that if when we're studying autism, if we don't restrict the study to just the affected individuals in the family, but rather measure the level of affectation in all the family members and use all the information from their phenotypes and their genetic background, it actually ends up increasing statistical power to find genes. And this is a demonstration of principle of that was published by Jennifer Lowe and our colleagues at UCLA just a year ago in the American Journal of Psychiatry. And this was a second demonstration of how that could happen if you incorporated really all the information. So essentially paying attention to the broader autism phenotype in that sense enhances statistical power for gene discovery. And keeping in mind that gene discovery, even though this is a very important target and an important end game for a lot of the research that we do, I just want to make sure that I emphasize the point that obviously the way genes operate is to influence cells and circuits and brain and behavior and a sort of a hierarchy of function that we believe that if we can get to the base of it, if we can contribute to understanding how genetic factors influence these outcomes, that we can have opportunities, not just at the level of genetic variation, but at the level of how they operate in cellular activities and circuits and various ways in which brain functions to intervene at any of these different levels once we understand how the system really works. So I want to now turn attention to just a couple of other points about the broader phenotype to sort of establish, I think, some major principles that we've learned from this. And then we're going to turn this over to more of a discussion. But I want to take you through just a few studies that really help to kind of cement what I think are the really important implications of this tale of three distributions. This particular slide depicts an intergenerational study that shows distinct shifts in the quantitative trait distributions for mothers and for fathers of children who have autism versus mothers and fathers of children who are typically developing. And so traces of the autistic syndrome are visible in these subtle shifts in the distribution across the whole population of these parents and indicate that these sort of aggregations that are contributing to autism risk are not only traceable in the prior generation, but this interesting fact that because the correlation between parents for these particular traits is very considerable, what happens is that when both parents are in the upper quintile of the distribution, so not affected, they're normal parents, but when both parents are in the upper fifth of the distribution for autistic traits and symptoms, it essentially doubles the relative risk for a full clinical diagnosis of autism in their offspring. And this was a study that was led by Kristen Lyall and her colleagues in the Nurses Health 2 study, which is a very large epidemiologic study. And this relates to a sort of principle that came into an even higher degree of focus when David Ledbetter and colleagues started to think about why it was that there was so much heterogeneity, even with particular highly deleterious variants. Here's the 22q11.2 deletion and the 16p11.2 deletion. These are chromosomal rearrangements that singly have been associated with, respectively, neuropsychiatric, an array of neuropsychiatric syndromes, and 16p11.2 is one of the most common chromosomal rearrangements that confer risk for autism. And it turns out that the individuals who have these conditions, so here's 22q11.2, here's 16p11.2 for intelligence, they have this wide range of affectation that the mutation doesn't determine exactly where they fall, and some of the individuals, are within the range of normality for IQ with each of these particular mutations that have been associated with these conditions. And Ledbetter and colleagues were thinking about, well, why would that happen? And they started to look at the parental distributions, and here are the parents of our general population and the parents of individuals with 16p11.2, and these are de novo mutations, so these parents don't have the mutation, but they show this very wide range themselves. And so the investigators thought, well, maybe where you fall when you have the mutation is not just a function of the mutation, but it superimposes on where your parental background lies. And in fact, this is what they showed for 16p11.2. And here are the results for not only for IQ, but for autistic trait burden and obesity as related to body mass index. And what they observed was that essentially the mutation was responsible not for your exact place on the distribution, but essentially for a shift in relation to what would be expected on the basis of where your parents or your relatives are in this distribution. And this is a very powerful demonstration of what the broader phenotype really means. So imagine being a child with 16p11.2 deletion, and your outcome, your life outcome, depends not so much just on that deletion, but where within the normal range your parents fell in this particular distribution. All of these parents are in the normal range. And yet, if they were in the low end of the normal range, what it would predict is that their offspring are on the low end of the normal range for the whole distribution for 16p11.2. So the idea that these sort of subtle background effects could have these profound influences on either risk or resilience in the setting of a mutation really has revolutionized our idea of what is a mutation in the first place, and the idea that it always operates against a genetic background and produces a predictable shift in relation to that genetic background, making it very pivotal what is that genetic background upon which it is superimposed. So that's a very important point. And in other kinds of monogenic syndromes, this is an example of neurofibromatosis. You can see other reasons for a whole range of outcomes in the patients. In this case, the mutation itself can be very different among different patients with neurofibromatosis, and the degree of autistic impairment that one family member has with an inherited version of neurofibromatosis is very similar to what their other family members have, indicating that for mutations in this particular gene, there are specific degrees to which they shift a person against their otherwise genetic background that's related to which mutation do they actually have. So we're starting to understand at the level both of the specific mutation and at the level of genetic background a whole different way of thinking about what does even a single mutation mean in influencing a condition. Another very important take-home message from studying quantitative traits in the general population, and I'll just take a minute to talk about this, is that when one conducts factor analyses of autistic, you take all the autistic traits and symptoms that are defined by these kind of checklist measurements, and you look at the degree to which in whole populations, so whether affected or unaffected, to which those individual symptoms travel together, you see something that's quite remarkable. So autism is defined by a clustering of social communicative deficits and deficits in or symptoms in repetitive behavior and restrictive interests. But it turns out in the entire general population that these travel together as well. And I have always felt that this means something very important about the biology of autism. We still don't have solid, robust, highly replicated neural signature for autism. We don't know where is the lesion. And I believe that the fact that social communication, which is one thing, and restricted interest in repetitive behaviors, including motor stereotypies, which is another thing, the fact that the intensity of those not only are correlated by definition in autism, but throughout the entire general population means that to me, the identification of a neural signature, finding the lesion, will be advanced by understanding how it is that some neural deficit could simultaneously produce variation across all of humanity in both of these things simultaneously. And I think whatever lesion is going to be identified, or the way it's identified, is going to recapitulate this biological truth. And what we do in our collaborations with folks in neuroimaging and studying neural circuitry in autism is to continuously raise this issue and to keep everyone vigilant about the extent to which these particular traits, even though you can ferret them out and kind of separate them in very, very large samples as different components of the syndrome, their trait correlations still are extremely high. So these very high sample sizes allow you to find statistically significant differences between clusters of traits, but they all massively correlate really across all humanity. And that is also shared, I'm going to skip ahead to, with something that has only recently been recognized, that motor impairment travels with these social deficits and with these with proneness to restricted interest or repetitive behaviors. And again, thinking about where's the lesion that would place this kind of this type of impairment in fundamental ability to coordinate motor movement with the proneness to not make eye contact, to miss the forest for the trees, and to not engage other people socially is a really important aspect of understanding what does quantitative variation in these traits really mean. And this particular relationship is very strong. The correlation with quantitative autistic traits and motor impairments in families affected by autism is on the order of 0.60, which is very significant. Okay, I'm going to skip ahead for a moment, and I'm going to get to some of the earlier origins, and we'll kind of wrap up the talk with this particular series of slides. Our colleagues now at Emory, formerly at the Yale Child Study Center, Jones and Klin, published this seminal paper. This one I think was in 2013 in Nature, and showed this remarkable finding of years of work. Talk about sticking with a phenotype that is very important. And this is visual social engagement, and showing that early in life the eye gaze of children who are typically developing is differentiated in their trajectory from the eye gaze of children with autism spectrum disorders. And this is essentially the proportion of time that young children spend when eye-tracked, looking at eyes versus looking at mouths, and recognizing that over time, over the course of the development of an autistic syndrome, a young child tends to sort of trail off and spend less and less time looking at eyes. And this was an observation that was made in comparing, primarily in comparing the later-born infant siblings of children with autism, comparing those who developed normally from those who went on to develop autism. A very, very important early finding. And I'm going to spend the time talking about how this relates to the broad phenotype and quantitative traits in just a second. But I first want to show this slide, which a number of you may have seen, but I think relates to this issue of this theme of how it is that genes actually function in human development, and how they interact with or are correlated with specific environments. I think this is a very important theme about thinking about any inherited trait, in particular, and certainly any quantitative inherited trait. So this is a snapshot in time during one of the videos in which Klin and Jones have studied the eye gaze of individuals, children who are typically developing versus children with autism. And this is a moment in time in a video clip that's shown to these research subjects, where a little girl is opening and closing the door of a toy car, and the subject is watching this. And at this particular moment in time, this kid gives this kid the death stare because he's really frustrated and wants her to quit opening and closing the door. And at that particular moment in time, it turns out the typically developing toddlers, for the most part, are riveted to this boy's face and eyes and facial expression, but the toddlers with autism are primarily on the door handle. And the reason I like to show this particular slide is that it depicts this idea that if you take this one snapshot in time and you extrapolate it, one of the issues is that what we believe is a difference between these two groups that is on the basis of a genetic liability that this group has that this group doesn't have, essentially results in this very early time in life in a world view of the environment that looks like this for a typically developing toddler and looks like that for a toddler with autism. And imagine extrapolating that over minutes to hours to weeks to months of time, that the life of an individual with typical development is a completely different environment than the environmental exposure of an individual with autism on the basis of a genetic factor that takes their eyes out of the play of what is the most salient aspect of a social stimulus. And to talk about why we think that that is genetically influenced, we took this same particular paradigm and have recently applied it in the same way that we did with twins in the population for measurements using the SRS and related measures, but making these same kind of social visual engagement measurements in infants at 18 months, looking at twin 1's proportion of time spent looking at eyes, twin 2's time spent looking at eyes for identical twin pairs, non-identical twin pairs, and unrelated siblings. And the same theme emerges, that for the identical twins, whatever twin 1 is doing, whatever proportion of time that they're spending looking at eyes, that's what twin 2 is doing. And that particular tight correlation between the two, that's on the order of 0.9, drops for non-identical twins to about 0.35 to 0.4, and then even lower for unrelated siblings. So we believe that social visual engagement in this way is under stringent genetic control, and we'll hope to be publishing those findings soon. But the reason I bring them to this particular forum in a preliminary way is just to make the point that we think that these particular features that would determine this kind of derailment over the course of time is under very strong genetic control, but then subsequently the consequence of that is that the deficit gets reified by changes in the environmental exposure, essentially, that that child is led to on the basis of that particular change. And in the general population, note that this is a general population sample, so this is not autism or not autism. There's a very wide range of variation within the general population. And so we're hard at work thinking about how this particular axis of inherited influence interacts with other aspects of genetic liability to influence the development of an autistic syndrome, and hopefully more on that very soon. So that also relates to, I think, what many of you know are recent attempts to treat autism early, and many of those attempts involve trying to right the ship of this gene-environment correlation that I just depicted in those prior slides, that is to take a child at high risk and orient them to the social world in a way that would not come naturally to them, but through intervention rights that ship so that much more of their environmental exposure is what a typical environmental exposure would be. And to the extent that one can do that, the question is, will that rehabilitate that liability? And obviously this particular study, which was a very important open trial, was very promising and published a couple of years ago. And we hope for continued refinement and replication of this as early intervention attempts move forward. So, the last point that I want to make about quantitative trait burden relates to that bimodal distribution and also has a treatment kind of implication, which is that for sisters of children with autism, this fact that autism is only recurs 1/4 of the time that it recurs in boys does it recur in girls. It turns out that although sisters are relatively less likely to develop a clinical autistic syndrome, they're still almost 10 times more likely than the girls in the general population to carry autistic traits and symptoms at a very high level. And so what we believe is happening in girls on the basis of these distributions and measuring autistic trait burden in the sisters is that this 4-to-1 ratio in autism is really a manifestation of a fundamentally reduced penetrance of inherited liability in the girls. And the great thing about this, the remarkable thing about this, is that it must be operating across numerous diverse mechanisms of transmission. And if that's the case, if across all the different disparate pathways to autism, that girls are relatively protected so that most females are protected from most, but not all, forms of autism susceptibility, then honing in on what is it about a female that allows them across numerous different types of autism to protect themselves from developing the syndrome, this would be a very important biological clue that if we could understand the secret of that, that might be applicable to a very large proportion of the autism population, irrespective of the genetic pathway to causation. And so we're very interested in understanding particularly the biology. This is an example of an extended pedigree where many individuals are affected by a very deleterious mutation, but only the boys are affected by autism, and the girls are unscathed and at times transmitting the condition to their then affected sons. So we're looking for these patterns of transmission, and we're trying to understand at the moment what exactly it would be about the female sex and the biology of that early in life, before there's any hormonal differences between boys and girls, that it would result in this profound difference in the recurrence risk and the prevalence of autism in a condition that crops up at 18 months of life or earlier. And that's called the female protective effect. And we've done some initial work in that, but a long way to go, but I think it should be a very important focus of activity because I think it represents a major clue that if we really understood the biology of it, we might be able to understand how to treat perhaps 3/4 of all autistic syndromes. Okay, so I'm going to skip ahead to the end then and say what I think are clinical implications for autism diagnosis. I think diagnosis then, as you can see, becomes a much more unclear kind of construct when you think, well, family members can be partially affected. When a particular deleterious genetic influence actually superimposes on what level of background liability do you have within your family to determine whether you have a diagnosis or not. When the very distribution of autistic traits doesn't lend itself to a particular cutoff of who's affected and who's not, and when the offspring of an individual who's perfectly normal may actually be determined by how much genetic variation within the normal range they have for these particular traits could make or break their offspring as far as autism liability. It's kind of prompting a sort of revolution in diagnosis, and we have to really rethink how diagnosis is made or how we understand what the implications of diagnosis are. Not that it's not important to characterize the more severely affected individuals who have clinical, levels of impairment and adaptive functioning, and I think it's very important that we understand that the status of a diagnosis rests not only on the counting of symptoms, but on the degree of impairment that is incurred by that symptomatology. And I think DSM-5 has tried to take a more astute approach to that, and I anticipate that there will be changes as the molecular basis of autism becomes more and better elucidated. All right, so I went a little over time, but I'll stop there. I want to make sure that I acknowledge our collaborators in a lot of this work, particularly our collaborators at UCLA, at the Kennedy Krieger Institute in the Nurses Health 2 study, our faculty members and a scientist at Washington University in the Social Developmental Studies program that study the genetic epidemiology and the phenotypes of autism, and our colleagues at Emory University who we are collaborating with to take these measurements to a much earlier, much more fundamental level, currently at the level of moving from a behavioral phenotype to this fundamental aspect of visual social engagement and looking at that relationship from early in life forward. So I think that is the end of our slides. And I'll let the moderators kind of pick up from there and start the discussion.
Great. Thank you so much, Dr. Constantino. That was a really great presentation. So we've definitely generated some questions. So we're going to have Christina Cottle moderate those questions for you. So take it away, Christina.
Thanks, Carolyn. Thanks, John. So the first question we have, and I'm kind of breaking it up into kind of content areas. So the first question is about the concordance rate of ASD between monozygotic twins. And the question is the concordance rate of ASD between monozygotic twins the same for high and low functioning individuals?
And the answer to that would be yes, although sometimes in an identical twin pair, there is a mix of the two. So, for example, one twin may be a little bit higher functioning, another twin may be a little bit lower functioning on the basis of either non-shared environmental factors. It could be that what are called somatic mutations. Those are mutations that arise after those two twins are differentiated in utero a little bit later in development and in a subsequent 4-cell embryonic stage or perhaps beyond that a mutation occurs that affects the functioning of that individual. So there's wiggle room in the sort of overall extent to which any given set of genetic factors will translate into a behavioral phenotype. But it really is quite highly heritable. And so, yeah, across whether you can say, is the MZ twin concordance similar for really low functioning types of autistic syndromes, which tend to be more represented in de novo mutations, because a lot of times the really very severe forms will be caused by mutations that would never have been carried by a mother or father because they're so severe that that mother or father would not have reproduced if they had that severe of a mutation. But irrespective of whether the mutations or the genetic variants that produce a really severe phenotype are polygenic in nature or these really deleterious hits and de novo mutations, those MZ twin concordances are very similar, and you can move to any range of the population, and see that same thing. Now one thing that you have to be careful of, and this has been you're going to see this every once in a while in the literature, but if you place a really rigid cutoff for autism or non-autism, you can see where if these are distributed traits, and you could say, well, let's say I'm going to place my autism cutoff at some score. Let's say the score is 70, okay? And twin 1 has a score of 71, and twin 2 has a score of 69. Some people in a twin study will count that as discordance, but that's not an appropriate construct for discordance because you couldn't even tell the difference between that pair of twins. And so the imposition of an artificial line in a quantitative trait will falsely, essentially in some studies, result in reports of discordance because a sort of hard line has been drawn in the sand. Well, twin 1 is like this and twin 2 is like that. So what it does is it takes all those twins that are kind of in the range for that cutoff that are identical twins, and naturally, if you're in that range that's close to wherever that investigator decided that was the cutoff for autism, then they're going to straddle that particular cutoff. And so then they're going to say, oh, well, the concordance rate for autism is only 70% or only 80% or only a trait correlation of 0.8 or what have you. And that's really not an appropriate way to examine the data mathematically. We can go into how to do this in a way that allows you to have a sort of a sliding window of where you're putting your cutoff. But the fact of the matter is that no matter what level of symptomatology we're talking about, and this is also true for the opposite end of the spectrum, for a high degree of social facility and high capacity for social communication, MZ twin correlations are very, very similar.
Interesting, thank you. We have a couple other questions about measurement tools and strategies for measurement. So the first one is, although the broader autism phenotype is continually distributed, are there BAP ranges, so to speak, on commonly used measures of autism, such as the AQ and the SRS, that can be used to define the group for research purposes?
Absolutely. I mean, I think the best way to think about this is also the way that we study IQ and intellectual disability, and so one of the the most coherent ways to do that in a standardized scale is just to think about where do you want to place your cutoff or your range with respect to the percentile demarcations in a population. So again, right now we're saying you have to be at a level of symptom burden that on average relates to the 1 percentile extreme of this quantitative distribution in order to get a diagnosis of autism. Our current prevalence rates for autism, all comers, again, 4 times higher in boys and girls, but all comers is about 1% of the population. Now, for most other quantitative traits, let's take, height, or let's take weight, or blood pressure, or blood glucose level. Most of the time, we start to see a level of impairment at about the 2.5 percentile of the population distribution, because essentially that's 2 standard deviations from the mean. And when a person's functioning at whatever level, if it's short stature or overweight or glucose level or blood pressure elevation, is 2 standard deviations from the mean, essentially in the highly evolved state that we as organisms are, things start to go wrong, and a person becomes, somewhat adaptively challenged as far as that's concerned. And that's true, certainly, I think everybody's sort of agreed that that's true for intellectual functioning. So I personally think that we are ultimately going to need to think about, what about the kids that fall in that range from the 1 percentile extreme, to the 2.5 percentile extreme? And we're pretty confident that they have a level of burden that I think deserves and requires and may actually be more responsive to interventions across the life course than the children and individuals who are unfortunately beyond that 1st percentile extreme. So that's the first thing I would say about that. The second thing I want to say is that because this is a quantitative distribution in the human population, and because highly conserved elements of genetic variation, that is common variants, account for over 50% of the population burden for autistic traits and symptoms. This means that those particular traits, when not in an extreme situation of, a high aggregation, those genetic variants, those common elements of what at the extreme are liability to autism, may actually be very adaptive when they're at lower levels in that distribution. So for example, individuals who tend to spend a lot of time on certain details, are very detail-oriented, and are able to persist without exhausting motivation for sticking to highly detailed kind of tasks. Those particular attributes may be highly adaptive, may actually even be evolutionarily adaptive in ways that these particular aggregations of traits, for example, might occur in a manner that improves, if you go all the way back in evolution, that improves survival value in a sense of particular variants that would improve, let's say, hunting and gathering ability in males, while simultaneously preserving in females the rearing and nurturing functions of our earliest ancestors. And so what I wonder about with respect to seeing these kind of sex differences in outcomes as a result of the aggregations of these traits, and the kind of dividing line where females are relatively protected, may end up being a manifestation of the biology of how these common elements actually evolved, how they were preserved evolutionarily, how these particular variants can be adaptive for males and females are relatively unscathed, so that their abilities to function from an evolutionary perspective of rearing are not in a certain way damaged by the sort of social repercussions that we see from aggregations of these traits. And And to take it forward just another step, and to get out of early evolution and kind of survival on the savanna, it's very interesting. Also, David Skuse showed this in a very interesting analysis of data on intellectual ability and social communication difficulties in general in whole populations. That as intelligence increases, it turns out that when you get to higher and higher levels of intellectual functioning, supranormal intelligence, it turns out that in boys, that ends up being attached to a social cost in a way that girls seem relatively protected from. In other words, that high IQ boys are more likely to have a proclivity towards autistic traits than their counterparts as females with the same IQ. And that also, we have learned, and this based on data that was acquired very carefully in Fragile X syndrome by the team now at Stanford, which was that when you look at individuals with Fragile X syndrome and you compare apples to apples for the FMR protein, it turns out that for boys, every 20-point drop in IQ that you get in that syndrome is associated with a certain drop in your capacity for social communication that is in the girls, even for the same drop in IQ, it comes at much less of a cost in terms of social communicative abilities. So again, thinking about these themes that when we think about this from a whole population perspective, that where we place dividing lines between health and disease, pathology versus variation, neurotypical variation, where we even think about where might these traits be adaptive, we just don't know that. And I think so. I really appreciate the question because I think scientists should think very carefully, first of all, about this kind of lost population of children, because we're so focused on this kind of 1% autism prevalence on the basis of traditional diagnostic kinds of categorical instruments. And yet we know there's this quantitative distribution, and we know that when things are quantitatively distributed, that we know that there is impairment that's occurring at those milder, still at the 2.5 percentile kind of extreme of the distribution. That's 2.5 times more people that would get binned in the autism range than are currently being binned in autism right now. So let's think about them. And are we going to bin them ultimately in that range and help them and support them, or are we not? I think that's a very fundamental question. And then on the other side of the 2.5% dividing line, when does it count as a disease or a condition, or when does it really just help you, or when does it just make you interesting and unique? When does it result, even when it's not in the clinical range, that if you end up selecting a mate that's in the same range, that it's going to increase your child's likelihood of having one of these conditions in a very significant way? And if your child is unlucky enough to have a loss of function mutation in a brain gene, that the extent to which that loss of function mutation is going to have legs in offspring outcome may be very, very heavily influenced by where that person falls in the range. This is just a different way of thinking about what's the meaning of a quantitative phenotype, and that's what I hope everybody had a chance to get that idea, and again, that there's more to the broader autism phenotype than meets the eye.
Great, thank you. I want to we had a couple of questions about, we're running low on time here, and I want to get to some questions about your career development, but I want to stay on the female protective factor for a second, or effect, sure, because we had a couple of questions on that. But one of the questions is, how much of the female protective effect is due to the way autism is diagnosed, and then also, is there any component of the female protective effect that can be attributed to environmental or societal differences between genders? I mean, you've touched on that a little bit, but I was wondering if you can just expand a little bit more.
Those are fantastic questions. So there certainly is diagnostic bias in autism related to, oftentimes, to the behavioral overlay of a individual who has a certain amount of autistic trait burden. So the way this works is, at the level, if you compare boys and girls with the same level of quantitative autistic traits measured by these kind of validated population-based measures, and if you take, if you just take boys and girls that have an equal symptom burden, it is absolutely true that it is more often that boys will get diagnosed than girls. But that only explains part of the ratio, because the other part of the ratio is that it's much more common for boys than girls to have that level of trait burden in the first place. So there's kind of two layers to it. But it's a very astute question, and it is true that there's a diagnostic bias, but it in no way explains the entire biological kind of difference between boys and girls, especially at the tail of the distribution. As far as the environment is concerned, we are very interested in the possibility that what might distinguish girls and make them relatively bulletproof, as it were, that is, most girls being protected from most forms of autism, might indeed have something to do with the environment. And we saw this in our earliest twin study, that there was a trace of common environmental influence among females in the general population that we did not see among males in the general population. And that environmental influence was a common environmental influence that operated in the direction of promoting social competency in females in the general population. So in other words, the idea is that it may be that females are doing something with the information that they're getting from the environment. They're responding to the early rearing environment in some kind of a way that pulls them out of the trajectory for autistic impairment. So imagine that in all humanity, there are subtle differences in the way boys versus girls sort of, pick up and use social cues and respond to that. Imagine if that particular difference that girls have in their responsiveness to the environment, that runs across the entire human population, that somehow that is the key ingredient that allows them to override, as it were, a liability to autism. And we actually do have other evidence to suggest that there are girls who start off very early with syndromes that look like they're headed in the same direction as their brothers. And one of the manifestations of this that we found in the Ian National Volunteer Register, that a very large number of girls in that registry, sisters of boys with autism, who were unaffected clinically, did not have clinical diagnoses, we showed that they had a higher autistic trait burden, subclinical autistic trait burden than girls in the general population, or more commonly in aggregation. But the other thing about those unaffected girls is that there was a group of them, much higher than the population prevalence for this, that had speech and language delays as young children, including weird autistic qualities of their speech, which they later outgrew in ways that the boys didn't. The boys went on to progress to autism. So we actually think that there may be something going on in that window of time, during the second year of life, that girls normally do in the entire population, in the way that they respond to the environment, that may be the key ingredient, or one of the key ingredients, how they're assimilating the information of what they're learning, what they're experiencing, that relatively protects them. And I think that's a really exciting place to focus analysis and investigation on, what are girls doing? What is the biology of that protection? Now, it also may be true that there may be protective effects of female sex at a much more fundamental level. We know that brain genes are expressed differentially by sex in early development. And so thinking about normal sexual dimorphisms over the course of development is a way to get some traction on what are possible contenders for the sex differences that really allow girls to be protected. So those are the kinds of things that we're doing, and even at the cellular level, thinking about if there are differences in the biology of the way neurons work that are expressed differently, even at the cellular level, which is actually true for sexual dimorphisms in risk for cancer, if you can believe that. That if there are things that we can identify and target, that this would be potential grounds for novel treatment.
Great. We actually just had a comment about studying kind of the infant social environment and parenting, how it's not a well-studied factor right now, but that could be an area for growth. Before we switch over to the career development, I just want to ask a couple of questions about family members, and so we had a couple of questions about in familial risk and everything like that. One question was, do all immediate family members of a person with ASD have some form of BAP? And the other question is do parents ever express feelings of guilt due to the high heritable nature of ASD, and how do you console parents in this situation?
Yeah. So excellent questions. The answer to the first question is the degree to which additional family members are, carrying or affected by these either subclinical components is really truly unknown, because we've only been able to specify them in a behavioral trait kind of manner. Now we know that from the standpoint of behavioral traits, it depends on where you draw the line of what's an extra burden of behavioral involvement with a subclinical version of the autistic syndrome. The issue is that the entire distribution of boys in families affected by autism is shifted. So how we can identify any single boy and say this boy probably is carrying some component of that syndrome, the only way we can do it now is very crudely by saying, well, what is their quantitative autistic trait score? And even if it's normal, if it's a little elevated, it's possible that elevation is because their brother has autism. It's also possible, realize too, that in the general population there's a normal range, and a higher burden of autistic traits can just be normal, can just be part of normal. So ultimately we're going to have to study families and molecular traces that link to these particular traits. But one of the things that's different for girls is that girls really typically show themselves to either be solidly within the normal range, or there's this notch in the distribution, or we start to see some with, a significantly higher level of affectation than sisters usually have, and yet still a subclinical version. And those are the ones that we think are the most likely to be quote-unquote carriers or silent carriers of the condition. And we're very focused on studying them from a behavioral standpoint, from a brain and circuitry standpoint, because maybe they have compensatory factors that they'll show us that particular population of girls that will give us clues as to how we might treat this condition. And so we're still searching for ways to really be able to assign a knowledge on an individual level basis about risk. Certainly there are rare inherited variants that are traveling in families that are not just these, very polygenic collections, but like kind of a smoking gun for, that 50% or 60% of the cause of that child's condition was contributed by one single genetic variant. And if that genetic variant's traveling in the family, then, specifying whether in a family member they have that same kind of smoking gun is very important. And so when we find that a child in a family has autism that's associated with a de novo mutation, it's pretty unlikely that another family member, it's impossible that another family member will have exactly that same de novo mutation. So that's kind of a relief. But from the quartet study that I showed, we know that those de novo mutations are not the only players, and that family genetic background still is playing a role. And so just being in the family of a person with autism, puts siblings at a 20-fold higher risk for developing autism in general. And so specifying in any one case what's the risk, that's something that really we're trying to work on right now in intergenerational studies that are funded and are underway, but it's still going to take some time to sort that out. As far as the issue of sort of the guilt of transmission, there is no question that families are anguished over, the possibility that, a genetic variant came from, one line of the family or another. On the other hand, I think families are also relieved by being exonerated from the blame that gets placed on not only autistic syndromes, but, neurobehavioral syndromes in general, when those syndromes are traceable to a biological cause that nobody knew about, that no one wanted for anybody, that the family members obviously, of course, wouldn't have wished on anybody that they loved or cared about. And so, you see a range of reactions to families about this. I think so far what we've been hearing from the families predominantly, and this is just my own personal experience, is that they want to know more, that they think that greater knowledge in specifying what is going on, because autism has been such a mystery and so overwhelming and so difficult to make sense of where did it come from and what the risks are. I think in general the predominant feeling of families is that more knowledge is better, and that's the prevailing consensus. But that doesn't mean that it isn't a tremendous hardship for a family, and sometimes people overplay the presumed knowledge of how much variance can be attributed to a given identified genetic factor. Remember, we talked about that it's not just that mutation, but that a child may have, but what the background that it operates on, and we're still just learning about that. And if a family is under the impression that, oh, well, if I were to transmit this particular mutation, my son or daughter would be, wiped out by that mutation, well, that's not always true either. That there are resiliency characteristics and genetic offsets that are operating that, wouldn't make that a necessity. And so understanding how that matrix works in risk prediction is something that is very, very important and requires, a lot of sample size and a lot of investigation.
Great, thank you. So we've had so many questions, and I want to thank everyone for sending them in. Unfortunately, we're not going to get to all of them, but maybe we can figure out a way to have some of them answered later on. But I want to, in our remaining few minutes, switch to some questions about your career development. I think one thing that stands out to me most about you is that you have this kind of dual career where you are a clinician and a researcher. And I was wondering if you could just speak a little to how you decided to go that route, rather being just clinical or just research.
Yeah.
And just talk about your experience there.
Yeah. Well, I mean, I guess I would say in one way I would almost think of it as a hedge. I think the needs of the families that are affected by this condition in the here and now, before we will have enough time to develop higher impact therapies, which is the goal of research in my mind, for them. But the magnitude of the burden to human health and the repercussions on families and so forth, that especially when they do not have access to appropriate clinical care, which definitely there is not enough of, not enough access, not enough availability, and so forth, made me think that, maybe even in a selfish way, that at least one thing that I wanted to be able to say for my career in the end was that I at least applied what we knew might help some subset of families as a contribution that I wanted to make, because science is a long road and an uncertain road, and I actually have a tremendous amount of respect for scientists who go all in and believe in the scientific process, and even if they rack up a whole agenda of negative studies, or it doesn't lead to a treatment or what have you, which often can be the case, that they've predicated their career on clarifying what the next step should be, and we all have them to thank. And it is totally true, the whole thing of standing on one another's shoulders in that sense. So, but I have to say that part of my career that I kept within the realm of taking care of the patients was important in a lot of ways. First of all, it's very hard to keep your nose to the grindstone of studying quantitative autistic traits, to thinking about transmission patterns in families, to looking for ways to characterize subtle manifestations in the syndrome in a phenotypic measurement unless you're sitting with the patients. And one of the great joys of my career was, to be doing research, for hours or days or what have you, and walk into the clinic and take care of a set of children and families and test the ideas that came up, within the research realm. Does this apply? When I am sitting with this family, when I'm working with this family, when I am, looking at the degree of symptom burden, transmission pattern, the degree of impairment, in what happens as a result of a treatment, does this hold truth? And so in some ways, the clinical realm has been a great refiner of my research and thinking about what's the most appropriate next step to take, including, keeping moving, towards the translational end. How are we going to get to an intervention? How are we going to understand the biology of this condition? How are we going to find the lesion in autism, the neural signature for autism, and how can I contribute to that by careful and large-scale phenotypic measurement? It's kind of funny because a lot of times I would say that people don't think of the kind of measurement that we do in large populations as very careful. It's 30,000 feet and it only takes 15 minutes, which is kind of true. But as I think you hopefully came through from today, you can get a lot of information in 15 minutes that means a lot when you're studying populations and strategically selecting populations and understanding what it is that you're trying to track down. So that part of it has been very important. And I have to say that when I interact with the community, who have been just incredible. Like I've stayed in the same place now for close to 25 years of a research career, and the community has rallied around our research program in all kinds of ways. And part of their rallying has been with the understanding that our team, our program, the people that I work with, are not only studying the children but taking care of them. And I think that's cemented a very strong relationship between the participants, who have been very loyal and stepping up to participate in research, and the research mission. And honestly, most of the families don't even understand anything of the implications of the research in the way that I've described them or lay them out today. They have vague notions of that. Of course, we tell them this is what the end game is. This is why we're doing these exhaustive serial measurements over time and across all the family members in your family. They're focused on their child's condition, and when we get feedback from the community, oftentimes, it'll come in the form of, if I'm giving a talk in the community and somebody's introducing me, they don't even talk about any of the research or whatever that they've all contributed to, but they tell a story of I or one of the members of our team, stopping, spending time, taking care of a patient, calling into an individualized education plan, leaving no stone unturned to try to do anything we can to improve the adaptive functioning of a child that's saddled with one of these conditions. And I have to say that that's been a remarkably gratifying aspect of my career and a remarkably frustrating aspect of it, in the sense that it is in some ways catastrophic that we cannot do more for these patients in the field of medicine, psychology, and social services than what we are trying to do. In some ways, it's almost an embarrassment for some of the families. I feel that way for what little we can do for some of the families. And that part of it, too, also drives the research mission, gets me up in the morning, puts me back in the lab thinking we have to come up with a better answer or a better set of solutions for what the patients are encountered with. So that's I think has been the value of a kind of a dual career in that sense.
Awesome. I love that. Thank you. And one last question, and as quickly as possible, but you mentioned picking one thing and studying the heck out of it.
Yeah. That was Eric Kandel's quote.
Yeah. How did you find your one thing?
Well, so the one thing for us was how I found it. It was totally by I backed into autism research. I have a separate line of research. I've always been interested in social development in children and from the beginning, but I started off studying violent and aggressive behavior and antisocial rather than asocial patterns of development. And long story short, backed into autism research on the basis of a grant opportunity to develop a new measurement schema for quantitative autistic traits. But the reason why in the beginning I thought that was important was because when I was taking care of patients, the teachers and the parents and the doctors were all disagreeing about who had autism. And so we would see a kid in the clinic that had a mild but significant autistic syndrome, and we would say to the schools, oh, well, let's help that child. Let's give that child an individualized education plan. And the schools would say, well, we're sorry, Dr. Constantino, but that child doesn't meet an educational diagnostic criteria for autism, and so therefore we're not going to give them a specialized education plan. This was monumentally frustrating to me. And when I combine that with the notion from Piven's work about the broader autism phenotype, and one of my mentors at a clinical attending at the Albert Einstein College of Medicine, who once wrote a paper entitled "Do Nerds Suffer from a Pervasive Developmental Disorder?" That was part of my upbringing, was this kind of collection of ideas that turned me towards, okay, let's get serious about measurement. Let's make measurements that everybody can agree on. And so the scales that we use can be actually, the ratings can be made not only by parents but by clinicians and by teachers, so that these ratings can be looked at across contexts. And when we were able to do that and do that in a way that was reliable across different contexts and pick the items that really made sense and differentiated these kind of populations, that's what kind of set us on our way. And then it became a matter of, well, we've got an index that seems to work. What are the implications of that index? What are the implications of looking at how that segregates itself in whole populations and with respect to inheritance? And then and that was how these iterations really arose in my career.
Well, thank you so much, Dr. Constantino. That was a great talk, but a great discussion. We're out of time. So I just want to end this session by thanking everybody who was involved, of course, Dr. Constantino, the working group, INSAR, and everyone for joining us live. I also would like to acknowledge Jennifer Gentry and Tessa O'Sullivan for all of their help with organizing these webinars. Video replays are available to INSAR members for the entire 6-course series, including this one. You can become an INSAR member by visiting autism-insar.org. If you have questions or want to get more involved, you can contact the Student and Trainee Committee at studentcommittee@insar-autism.org. We hope that you've enjoyed this session of the 2016 INSAR Summer Institute series. Please take a few moments to leave us some feedback in the comments window at the end of your Ready Talk session. We plan on holding these webinars again next summer, so stay tuned for more information throughout the year. Thank you again so much for joining us today.